Welcome to your first genetics lab, DNA extraction. We will use these videos to both provide you with a list of materials that you're going to be using in the lab and show you the procedures that you're going to be using in the lab. You'll need a 100 to 1000 microliter pipette for this lab, a 10 to 100 microliter pipette, the pipette tips for the larger and smaller pipettes, they're color-coded. You'll also need a bunch of weigh boats and prods. In this lab, you'll be required to use razor blades. To get them out, you push with your finger on the exposed blade and can remove it. You'll have a reagent kit with resuspension solution, lysis solution, neutralization solution, matrix, wash, and plain water. You'll also need a bunch of clean microtubes and 2 spin columns. Before we go any further, you need to know how to use a pipette. Make sure that you are familiar with the 2 stops of the pipette. Attention! When putting a tip on the pipette, never touch the tips yourself. When using a pipette, press to the first stop before you go into a liquid, and then release the button to fill. When releasing the liquid, push all the way to the second stop. Additionally, you may have to mix using a pipette. To do this, press in and out to the first stop to mix. Always replace your tips between use by using the eject button on the back of the pipette. For the first part of our DNA extraction, we will need a tube that's labeled 1 and 2, as well as our fish sample. We will use the weigh boat in order to measure our fish sample. At the weigh station, first zero your weigh boat. Next, cut off a piece of fish to weigh. You may instead get prepared fish in ethanol of the correct size, roughly the size of a pencil eraser. We were looking for about 100 milligrams of fish, so this was definitely a little too much. Once you've got about 100 mgs, you're good to go. Now we have a new weigh boat and a new pair of gloves. We're going to cut up our piece of fish as small as we can get it into mincemeat. Once your fish is small enough, add it directly to your test tubes with either a 1 or a 2 written on it. You will then repeat this process with your second fish. A used razor can be slid into the back of the razor container, where it won't be able to come out until the razor container is thrown away. Now we will use our bigger 100 to 1000 microliter pipette, along with the tips designated for it, to add 200 microliters of resuspension to your 2 minced fish tubes. Once you have added the resuspension fluid to the tube, flick the tubes in order to ensure that the fish is fully submerged. Discard your tips between fishes so as to avoid contamination. Next, we will add our lysis to our tube. Set your pipette to 250 microliters. Then add this to each tube. After adding the lysis, mix the tubes by inverting them 10 times. We will now incubate them at 55 degrees Celsius for 10 minutes. Once the incubation is done, we will add 250 microliters of neutralization to each microcentrifuge tube, and then mix by inverting. This is done, take your samples to the centrifuge to be centrifuged for 5 minutes at 12,000 Gs. Make sure that you always exactly balance the centrifuge to keep it from going off kilter. Add the internal lid and close the top before you attempt to start the centrifuge. When your time is done, press the button to open the centrifuge, then use the buttons on the cap to open the internal lid. Remove your samples and continue with the experiment. Make sure that you close the centrifuge again before leaving it. Next, we will use our spin columns and snap the bottoms off of them. Then insert each into a 2 milliliter microcentrifuge tube. Label these 1 and 2 for your 2 different fish samples. We will transfer all of the liquid that we just centrifuged into these spin columns. Next, we thoroughly homogenize our matrix. We're doing this by using a vortex machine. Now, we will use our large pipette to add 200 microliters of the matrix to the first column containing fish extract, and then pipette up and down to mix. This is what that mixing looks like. Try not to add bubbles. Repeat this process for your second fish sample. Now we will centrifuge our samples in the spin columns again at the highest spin speed for 30 seconds. As the centrifuge does not have a 30-second setting, you will have to stop the centrifuge once 30 seconds have elapsed. Now, take your samples and pour out the flow-through in the bottom tube into the sink. Now we will add 500 microliters of our wash, labeled W, into each of our tubes. Centrifuge for 30 seconds at top speed again. If you need to turn on the centrifuge, you can use its button, which is located on the back right above the power cord. Pour it out, add 500 more of wash, then repeat again. After completing your second wash, then centrifuge the columns again for 2 minutes. While centrifuge is going, label 2 new clean microcentrifuge tubes with your fish sample names. Place our spin columns full of dry matrix into these new labeled tubes. Using a pipette, add 100 microliters of the distilled water labeled H2O to each spin column. We will now take these back to the centrifuge, where they will be centrifuged at full speed for 1 minute. Because I used capless tubes, I now have to transfer the eluted DNA, the clear jelly-like stuff in the bottom, into another tube labeled with my initials and the fish sample. If you instead use a capped tube, you don't have to do any of this transferring at all. Once your sample is done, close it and hand it to your TA. It'll be stored at 4 degrees centigrade until your next lab.